Hey all, so we're going to be looking at D4.1, which is related to natural selection. This is HL content, and we're going to be looking at page 749 to 756. We're thinking about the theme of continuity and change, and the questions we want to be able to answer at the end of this are what processes can cause changes in allele frequency within a population, and what is the role of reproduction in the process of natural selection. So when we're thinking about gene pools, first we need to think, what is a population? That's a group of organisms of the same species living in the same area. And a gene pool is all the different genes and variations, the alleles in a population that are contributing to reproduction. Fitter individuals are going to contribute more to the gene pool. And equilibrium exists when all have an equal chance of contributing to the gene pool. So separate gene pools may exist when populations of species have been separated via geography, and while they could potentially interbreed, they don't. Allele frequency in humans differs by ethnic groups, or can differ by ethnic groups, as well as geographic regions. And so on Alfred, which is a Yale database, you can see this. And an example of this would be the allele for sickle cell anemia. And so populations of humans who arose in Central and Western Africa tend to be more likely to be carriers or have sickle cell anemia or sickle cell trait because it confers resistance to malaria. And so in this area, it was beneficial, advantageous for survival and then reproductive fitness for you to have one allele of sickle cell, because it conveys resistance to malaria. And so we can see this arising in other areas as well. Obviously, these would be successful in areas with malaria and without medical intervention like we potentially have now in most areas. But this could have arisen separately, or alternatively, it could be a byproduct of migration of humans. So when we're thinking about changes in allele frequency, firstly, we need to remember Gregor Mendel. He's the guy, Mendelian genetics guy, who did the pea plants experiments. He showed transmission of heredity using some kind of unit in 1866, and this is the gene. And then August Weismann had his germ plasm theory in 1892, saying that acquired traits can't be passed on from somatic cells to offspring, because that would be, there's just no functional way for body cell tagging to be transferred to your reproductive cells, your gametes. If you have grown muscle, for example, that's not going to be passed on to your offspring. Your offspring could grow their muscles, but they're not getting that directly from you. And so neo-Darwinism, post-Darwinism, is considering Mendel as the father of genetics, Darwin, the father of natural selection and evolution, and then Weismann considering, acquired traits can't be passed on. So there's three patterns of natural selection. We have stabilizing selection, which is where the middle of the road gets selected for, and you don't have extreme options like peacocks, tiny tails, or massive tails. You only have a middle-sized tail. And then we have disruptive selection, where we're going to remove intermediates. And so you would have black mice or you would have brown mice, but you don't have any in the middle. And sometimes disruptive selection is also called diversifying selection. And then we have directional selection, which is where one trait gets selected out over the other. And so we saw this with the moths. Initially, they were white, but because the trees got dirty due to coal dust, most of them became black because they were better able to survive on the dirty trees. So it's going to push for one extreme. Artificial selection of crops has resulted in directional selection for larger varieties. And we can see this in tomatoes that used to originally just be like cherry tomato sized, corn, which used to be a type of grass, and then wild pigs were like a mid-sized dog size, but now the pigs that we grow are quite a lot bigger. So Hardy Weinberg, this is going to tell us the likelihood of the frequency of certain traits occurring in a population. And so when we are thinking about this, we have albinism. So for example, is occurring in 1 in 20,000 people. And so Q is going to be the recessive gene here, so frequency of homozygous recessive genotype, and then P is going to be the frequency of homozygous dominant genotype. And so if we are thinking about this, Q squared is going to be, well, Q is going to be 1 over the square root of 20,000. And Q squared is going to be 20,000. And so that's going to give us a frequency of 0.07. And because P plus Q equals 1, we are going to end up with P being 0.993. So then the probability of the homozygous dominant genotype is P squared, and P squared is going to be 0.986. So we have a much more likely probability of P squared occurring than Q squared. And the probability of the heterozygous genotype is going to be 2 times P times Q. And so if we do that, we're going to end up with a value of 0.0139. So this process to figure out the likelihood of this occurring, or the allele frequency of this, is assuming that there's no mutations in the genes, that mating is random, there's no immigration or emigration. That should say emigration. The population is large, and natural selection is not favoring one phenotype. And if the frequencies are following that, like, so you can see this happening in using this formula with the population, then there's a genetic equilibrium. But if the frequencies that are being calculated are not following this, what's happening, assuming that the other 4 characteristics here, population is large, there's no immigration or emigration, mating's random, and there are no mutations in the genes, then what's happening is natural selection is favoring one phenotype. So to summarize, actually just one quick second here with Hardy-Weinberg, it's likely that something like this would probably come up on an exam. They like to give some kinds of calculation formulas, so you probably would want to know this. So a gene pool is all the different gene alleles in a population. This can vary within a species according to geography due to selection pressures, and they may have separate non-mixing gene pools. Weissman's germplasm theory, Darwin, which said that your somatic cells don't transfer your acquired traits to offspring. Darwin had the theory of evolution based on natural selection, and then Mendel, he had genes are the units of transfer for genetic traits. We have natural selection is going to be either stabilizing, disruptive, or directional. And the Hardy-Weinberg equation can allow us to calculate the frequency of a trait in a population and the probability of an individual having that gene combination. Artificial selection has allowed for directional selection of crops. And that's it for this video. So thank you very much for watching.